Welcome to Quicksand, the most important news in pharma and biotech this week. The FDA finalized guidance stating it does not intend to grant orphan drug designation for pediatric subpopulations of common diseases in an effort to close a loophole in the Pediatric Research Equity Act, or PREA. While PREA was enacted to promote pediatric research, the act included an exemption from pediatric study requirements for orphan designated drugs. This exemption created a loophole that allowed sponsors to get a pediatric subpopulation designation for a non-orphan disease in adult populations and be exempted from conducting pediatric studies normally required under PREA when applying for an adult indication for that disease. The final guidance comes less than a year after FDA Commissioner Scott Gottlieb announced the agency would work to close the loophole, and 7 months after the draft version of the guidance was released for comment. FDA does not expect to grant any additional orphan drug designation to drugs for pediatric subpopulations of common diseases, FDA writes. However, FDA says it may continue to grant orphan designations for pediatric subsets of common diseases if it would not be appropriate to use the drug in the adult population or if the disease is in fact a different disease from the disease in the adult population. The FDA published a list of surrogate endpoints to help inform drug developer discussions with relevant Center for Biologics Evaluation and Research or Center for Drug Evaluation and Research Review divisions. The list, which was created thanks to the 21st Century Cures Act, includes surrogate endpoints that sponsors have used as primary efficacy clinical trial endpoints for approval of new drug applications or biologic license applications. It also includes surrogate endpoints that may be appropriate for use as primary efficacy clinical trial endpoints for drug or biologic approvals, although they have not yet been used to support an approved NDA or BLA. The list, which will be updated every 6 months, features surrogate endpoints for numerous diseases, including acromegaly, different cancers, chronic kidney disease, cystic fibrosis, hepatitis A, B, and C, HIV, hypertension, and osteoporosis, among others. The list, which separates adult and pediatric endpoints, does not include composite endpoints that are a combination of biomarker surrogate endpoints and clinical endpoints. The table also does not include surrogate endpoints that may have been accepted for past drug development programs but are no longer acceptable as an endpoint to support registration. The FDA released draft guidance to help sponsors understand the evidence necessary to demonstrate the effectiveness of new drugs, including biologics, or new drug uses intended for slowly progressive, low-prevalence rare diseases that are associated with substrate deposition and are caused by single enzyme defects. In addition to the recommendation that sponsors perform genetic testing for the defects of interest in all clinical trial subjects, FDA explains how, in the absence of a way to directly characterize the clinical response to the drug of interest, the non-clinical and, in particular, the clinical pharmacology components of the drug development program become the main source of data that support a safe dose that can be used to initiate human studies and inform dose exploration, which is essential to final dose selection for clinical trials. Animal models, FDA says, can provide evidence of enzyme activity by demonstrating the reduction or disappearance of disease-specific substrates. FDA calls on sponsors to discuss plans to generate evidence of substrate reduction in clinical trials. Such evidence should be generated in tissues where changes in substrate deposition can be readily measured, and the relevance of changes in these tissues to the overall disease process must be well understood and clearly justified. The sponsor should also address how the treatment effect size relates to the variability in the test measure, the draft says. And FDA notes that this guidance does not apply to low-prevalent rare diseases with rapidly progressive clinical courses, as such conditions can be evaluated by traditional approaches, and low-prevalence rare diseases with previously characterized endpoints predictive of clinical benefit. The FDA released new draft guidance on recommendations for optimizing and standardizing dietary management in clinical trials for developing drugs to treat inborn errors of metabolism, for which dietary management is a key component of metabolic control. The 8-page draft discusses how sponsors should consider stabilizing patients' diets across sites to ensure consistency. A trial's inadequate dietary management can make interpretation of results particularly difficult when the average treatment effect of the new drug product is relatively modest, FDA says. Measurements of concentrations of metabolites primarily in serum and urine are often used to inform dietary changes and to determine whether dietary management has been optimized. Sponsors should select the duration of the pretrial period for optimizing and stabilizing a patient's diet based on the specific condition that is being treated, the severity of the metabolic defect, the patient's age, and the complexity of the dietary management, the draft says. It also calls on patients to enter the trial during a period of metabolic control, as demonstrated by appropriate biochemical analytes. Baseline dietary information should be documented over an appropriate time period at enrollment, even if metabolic stability has been shown. The draft also discusses planned procedures such as surgeries, clinical trial design, including considerations for optimizing diet, challenges and limitations of diet assessments, and use of historical controls. The European Medicines Agency adopted revised versions of this drug development guidelines for two inflammatory bowel diseases, Crohn's disease and ulcerative colitis. Last updated in 2016, the 19-page guidance on Crohn's disease provides recommendations for the clinical development of drugs to treat the condition, including patient enrollment, endpoint selection, study design, safety considerations, and risk management plans. For its second revision, EMA says the guideline has been updated with changes to the recommendations for study design for adult patients with regard to treatment claims, primary and secondary endpoints, and comparators to use in the studies. According to EMA, sponsors should no longer use combined indexes such as the Crohn's Disease Activity Index as the primary endpoint for the studies. Instead, the agency says sponsors should evaluate symptoms and inflammation separately as co-primary endpoints. As for comparators, EMA says it is necessary to have a direct comparison against a standard first-line treatment. EMA also says that having a placebo arm is ideal unless the study aims to show superiority against a standard of care comparator. In its first revision to its 2008 guidance on developing new products to treat ulcerative colitis, EMA has updated the 18-page guideline with new recommendations for study design, endpoint selection, and possible comparators. As for primary endpoints, EMA says that sponsors should measure both the proportion of patients with symptomatic remission based on patient-reported outcomes and the proportion of patients with endoscopic remission. EMA also lays out similar criteria for comparators as in the Crohn's disease guideline, noting that a direct comparison with a generally accepted standard first-line treatment is necessary for first-line indications, while a placebo is acceptable for second-line indications. Thank you for listening to Quicksand, episode 40, July 30th, 2018.